We're going to get started with the first session, which is focused on cancer-specific considerations. And the moderator for this session is Dr. Ricky Peters. Dr. Peters is from the Fred Hutchinson Cancer Center in the University of Washington. So I'll turn it over to Dr. Peters.
Thank you. In the interest of time, I will keep my introductions very short. The first speaker of our session is Dr. Mary Beth Terry from University of Columbia University, Mailman School of Public Health. Thank you, Mary, for presenting.
Wonderful. Thank you so much. And I'd like to thank the organizers at NIEHS and NCI for their vision and hard work in putting this conference together, and to thank all of you for joining today. I was asked to give a brief overview on the state of the science with respect to epidemiology and how we produce rigorous evidence evaluating environmental exposures, which I will broadly represent as E throughout this talk. I hope by the end of this talk, you will agree that study design is essential to think about when making inferences, particularly in studies of diseases like cancer with long induction times, and that most importantly, all samples are not equal. And just having samples for omics research without the context of when they were collected and in whom will lead to underpowered and biased research results. No conflicts of interest to declare.
Mary, I don't think your slides are showing, or am I the only one who don't see them?
Yeah, I think you need to share your slides.
Oh.
Yes, we see them.
But you didn't wait till the end of the talk to tell me that. Okay, great. So as an epidemiologist, I like to start with absolute incidence and changes over time. Here we can see three major observations about cancer in the United States for the last 50 years. First, most cancers are diagnosed at age 70 and older. Second, the largest annual increase in cancer incidence are in adults under 55 years of age, particularly in women, where the rates under 55 years of age far surpass that of cancer in males. And third, there's the power of risk reduction at a population level when exposures change over time, as you can see here with the decline in cancer in older males, primarily driven by reductions in smoking. It's like incidence in males over 70 are also coming down across all racial and ethnic groups. But it's critical to emphasize the major differences in the starting points. The implication of these different starting points means that in order to meet the cancer moonshot, which calls for halving the cancer mortality rate in 25 years, translates into a much larger decline in some groups compared to others if we are to reach this goal for everyone. As you can aim for the cancer mortality moonshot of 73 cancer deaths per 100,000, it would translate into a reduction in mortality of almost 61% in African Americans compared to about a 26% reduction in the mortality rate for Asian and Pacific Islanders. For context, if we only aim for the average, we will still have major health inequities. For example, with the CDC Healthy People 2020 cancer mortality goals, the metric was to reach 160 cancer deaths per 100,000, represented by the gray bar by 2020. This goal was met, I know both from the data and from Twitter, but it was met only on average. As you can see, it wasn't met for African Americans. Can we ever say that a goal is met if it's not met for everyone? To achieve health equity, we need to ensure the goal is met for each and every strata. We need to move beyond the average. 5 brief points. First, we need to move beyond evidence-based medicine hierarchy. A hallmark of evidence-based medicine hierarchy is that randomized controlled trials are specifically given the most weight in forming clinical guidelines. Yet we know that there's always a trade-off between inference and generalizability. Designs for public health impact, policy, and evaluations prioritize generalizability and generally, as a result, favor population-based sampling, where RCTs have always incorporated more targeted sampling. Further, we know that most cancer causes have been identified from observational research, not from RCTs. Within observational research, population-based studies have been given preference over non-population-based studies, likely because of this importance of generalizability for public health purposes. Here it is about moving the average in a population. However, what I hope to convince you of in this brief overview is that population-based cancer research studies will be underpowered to detect gene-environment interactions, as most cancers are identified much later in life. And more targeted sampling, ensuring that there is enough power in each strata for research on omics, will be needed for valid inferences. We need to move beyond the average. The old paradigm for cancer risk separated the majority of cancers that occurred in the population as environmentally caused, and much observational research over the last half a century identified major causes of cancer, as you can see on the right. These studies were primarily conducted in unrelated individuals. The remaining cancers that clustered within families were thought to be driven primarily by germline genetics, and the identification of many of the hundreds of cancer genes started first with these family-based studies. What we now know, however, is that this was a false dichotomy. No cancer gene is 100% penetrant, meaning all cancers involve gene-environment interactions. This graphic illustrates some of the cancer genes that have been discovered and the different organs that alterations in these genes affect the most. The size of the gene indicates its population frequency. These genes are relevant to everyone and are altered through both germline and acquired mutations throughout life, not just relevant for people with a family history. But many genes needed an enriched study, like a family study, for identification. I hope that you see that enriched studies will help identify environmental factors as well. Illustrates the majority of the cancer hallmark genes, about 1,200, can be affected either through the germline, represented in pink, or somatic level, represented in yellow, meaning altered throughout life. Cancers on average harbor 13 germline functional variants, but again, we want to move beyond the average, and the range is quite wide, 4 to 29, but on average with somatic mutations, it's about 23, with a range of 0 to over 3,500 somatic mutations in cancer hallmark genes. On the right, you can see the clear trends in the proportion of germline to somatic mutations across life, with the germline burden decreasing as age increases and the somatic burden increasing. This reality calls for much greater attention to study design and moving beyond this hierarchy of evidence-based medicine. And specifically, if there's questions about germline variants, these studies should be conducted earlier in life, and studies recruiting older individuals need to think more about acquired mutational burden. Conducting studies of germline variants in older individuals without factoring in other susceptibility could lead to biased results. I'd like to briefly discuss the role of omics to introduce the different platforms that will be discussed across the 2 days. Throughout this conference, we will hear about 5 major types of omic platforms. From left to right, we can think of the different omics platforms as a progression from DNA, DNA readout, RNA, protein, metabolites, and their role in epidemiology study design will be informed by when they are measured with respect to exposure and how they are measured, whether in peripheral tissue or in the tumor tissue. Epidemiologic studies of cancer have mainly focused primarily on genomics and to a lesser extent epigenomics, but are starting to collect samples in a way that can allow for measurement of other omics. But even as the technology improves to make it possible to measure different omics platforms, it is essential to think about the timing of when they are measured. Genomics and other omics platforms, if measured before the exposure occurs, can be used to understand underlying susceptibility. In other words, as a modifier or an interaction term. If measured after the exposure, all of the omics platforms except germline genomics may be used to evaluate potential mediating pathways and/or as outcomes for an intervention study. For one could measure metabolomics, for example, before and after an intervention to reduce exposure. Increasingly, genomic instruments using germline variants are being used as proxies for exposure to reduce biases seen in observational research using a method called Mendelian randomization, which uses genetic variants as a proxy for exposures, as these proxies should not be confounded given Mendelian genetics. However, even though you can measure genomics at any time, how you interpret inferences from genomic studies will very much depend on how and when you sample, and studies using samples from older individuals will likely be biased by survivorship of who remains in the populations and joins the cohort at the time of the biospecimen sampling. study design, but sampling and specifically who and participants join a study that is crucial for inference. Here on the right, we see how fundamental research is overrepresented by white individuals and very much underrepresented in most other racial and ethnic groups. This is a major gap in drug development tested in clinical trials, which have been important at reducing cancer mortality, is conducted with samples that very much do not mirror the global population that you see on the left. I'd like to then give an example to further consider why we need to move beyond the average and ensure stratum-specific statistical power is sufficient. I'll provide the example with breast cancer, which is the most common cancer globally in women. And most studies of the environment do not factor in susceptibility, including windows of susceptibility, where we know the breast tissue is more vulnerable to environmental exposures, as this is when the tissue is rapidly changing in form and function. And so having these studies conducted outside of consideration of susceptibility has led to a lot of inconsistent results. When you look closer at studies that have factored in susceptibility, you can see that the story is very different. To illustrate why knowing about susceptibility is so crucial, we conducted a systematic review of many common environmental exposures and breast cancer. The different ones are listed on the left. The type of enriched studies are reflected based on the column and whether they considered enrichment in the design or the analyses. The study types, including family-based studies, early onset cancer, and stratum-specific results based on cancer-causing variants. The color indicates the number of studies conducted. And what you can see is most of these exposures on the left had inconsistent evidence with regard to breast cancer risk in the epidemiological literature, again, which is largely based on average risk women. However, when you examine the 68 studies that did factor in susceptibility, 80% of the studies conducted in women with a breast cancer family history, 80% of the early onset breast cancer studies, and 74% of the gene environment interaction studies were statistically significantly positively related to breast cancer for the highlighted chemicals. Thus, when factoring in susceptibility, the evidence is much more consistent. But just like identifying genes in enriched studies that have relevance for all women, these findings indicate that environmental chemicals may affect risk in average risk women, but that the studies are underpowered to detect them outside of an occupational setting. Using more of a targeted sampling approach, I show the distribution of absolute lifetime risk in our family cohorts, estimated based on family cancer pedigrees and known genetic susceptibility genes. Here you can see that the distribution of absolute risk of breast cancer on the x-axis has a wide range compared to what you would see in an average risk cohort. In pink, you can see that a family-based cohort has sufficient power to estimate stratum-specific effects at the tail of the distribution, just like with genes, and therefore has greater power to detect gene-environment interactions. But a family cohort also has a sufficient number of people who are at average risk as well, so that there's sufficient power here represented in blue. The variation in absolute risk is much different in average risk cohorts represented here in blue compared to enriched cohorts. And as a result, average risk cohorts will be underpowered to detect gene environment based on germline genomics. Enriched cohorts not only offer more power for gene environment interactions, but have much larger range of absolute risk based on germline genetics. They also may be able to detect stronger signals from common ubiquitous environmental exposures. Here we illustrate the concept with our family-based study, where again, the predicted absolute risk is on the x-axis and the estimate of the association between polycyclic aromatic hydrocarbons, which is also referred to as PAHs. The protein adducts here are measured in blood with respect to breast cancer. For individuals that have high level of PAH protein adducts, compared to individuals without detectable levels, the odds ratios were all over twofold and increased to over sixfold based on absolute risk. In this range, it is very unlikely that bias can explain away the association. Compare this to what would happen at the lower end of the absolute risk scale, where it remains possible for bias to explain away the association, and that's represented on the lower left quadrant. So I'd like to then discuss this concept of life course and why windows of susceptibility is important. So twin studies have provided even more evidence that DNA is not your destiny and what happens across life matters. The largest twin study, using data from approximately 200,000 twin sets, supports that risk is much more than the genes. And you can see here that even if your identical twin was diagnosed with breast cancer, you have less than a 30% cumulative lifetime risk that you will have breast cancer. Thus, the much more likely scenario is at 70% is that this identical twin will not get breast cancer. Similarly, you can see for any cancer, if your identical twin had a 46% lifetime risk of having any cancer, but a 54% cumulative risk of not having any. We need to know what modifies the non-modifiable, which undoubtedly includes both endogenous processes and exogenous exposures. Endogenous aging processes and environmental exposures that get under our skin change over the life course, which is exactly why susceptibility matters, and measurement of susceptibility over the life course is essential for understanding cancer etiology. There's a direct relationship between cancer incident curve in red and the aging curve in green. However, unlike this direct relationship, the somatic mutational burden, which we know increases across life, does not directly align. In fact, roughly half of all mutations and epigenetic changes are estimated to occur before the full body maturation. The accumulation of genetic and epigenetic changes significantly slows later, following by a deceleration of stem cell division rates when stem cell transition from body maturation to body maintenance. Thus, before the cancer incident curve really accelerates, half of the mutations that influence cancer risk have occurred. Yet another reason why cancer risk reduction needs to start much earlier in life. Mutational burden and the fact that breast tissue changes during specific periods of life, including the intrauterine period, pubertal window, pregnancy, lactation, menopause. The vast majority of epidemiological studies on environment and breast cancer occur outside of these windows of susceptibility. The chemical classes are listed on the left. Here, the dark blue indicates at least 10 studies exist, where the yellow indicates only one study and the white indicates no study. Few studies exist that specifically address windows of susceptibility, and those that do are primarily animal studies. However, when we zoom in on the studies that do look at specific windows of susceptibility, the evidence is more consistent, just like when we think about underlying susceptibility based on family history or genetics. For example, we see that human studies of pesticide exposures during infancy and early childhood affect breast cancer risk, as do chemicals during pregnancy and menopausal windows. Susceptibility matters when measuring the effects of common environmental chemicals. I'd like to conclude by then just discussing some of the designs that may be useful as we move across the cancer control continuum. So when we think about etiology and primary prevention, I would argue that we need to think both about these higher risk cohorts that have more power to detect germline mutations earlier life, and that if we are going to have older cohorts, we definitely need to factor in susceptibility from acquired mutations and other alterations. However, if we are thinking about prevention, we still may want to look at population-based interventions. When we're thinking about cancer screening, again, more and more studies are evaluating risk-based screenings, but again, focusing on the important purpose of having cohorts to validate those models. And then finally, I would argue that case control studies may be very useful for understanding how therapeutics and prognosis change after a diagnosis. As you can think about collecting both tumor tissue as well as repeated measures of blood and urine after the diagnosis to understand how environmental chemicals affect therapy and treatment and prognosis. And also would allow for an opportunity to do cascade risk reduction with the family members. So, selecting cohorts to analyze, we do need to think about some of the limitations of many of the existing cohorts. First and foremost, many of the existing cohorts around the world are not diverse enough. They're definitely skewed towards older individuals where most cancer is diagnosed. That will definitely lower the statistical power for assessing germline genetics, but also, and perhaps even more importantly, enrolling into a cohort, we have what geneticists sometimes call deleting the susceptible. So you have to already be, to enroll in a cohort at baseline, is already a selection bias of who gets excluded. And so this very much applies to Mendelian randomization studies in cohorts of older individuals. So some solutions may be to establish new cohorts. You've heard about some of the new exciting cohorts funded by the NIH. And if we think about establishing new cohorts of younger, more diverse individuals enriched for cancer risk, and if using existing cohorts of older individuals, we definitely need to think more about measuring susceptibility beyond the germline genetics. So finally, we need to consider the impact of underlying susceptibility, particularly for common environmental exposures outside of an occupational setting. Population-based cohorts or cohorts that enroll most participants at midlife or later need to consider survivorship bias and need to focus on questions related to mutational burden. Estimated that half the mutational burden before the body matures argues for study designs that capture windows of susceptibility. And finally, oversampling groups underrepresented in omics research is key to reducing the cancer burden. So finally, I was asked to briefly summarize what the implications may be from what we know. So some regulatory and screening policies should continue or expand and do not need omics research to support these policies. These would include anti-tobacco policies, enhanced cancer screening based on occupational exposures, and expanded cancer screening to areas with high environmental contamination. Omics research will be important for other regulatory policies and decisions for EPA, NTP, and IR. And in particular, omics research is really critical to thinking about building the case for how the environment gets under the skin, finding and identifying higher risk individuals for cancer screening, understanding mechanisms for reducing risk from prior exposures. And central to study design choices and sampling must be the need to address cancer health inequities and reach the cancer moonshot for everyone. I'd like to thank you for listening, and I'm happy to take any questions or comments.
Thank you, Mary Beth. This is a very interesting presentation. There's one question that's specific to one of your slides in the chat that is asking that this complex slide that you showed where the cancer incidence goes up as age goes up, but the slide also shows that the mutational accumulate early in life and sort of top out later in life. Why would that be? Would the mutation be occurring throughout one's life?
Yes, no, it's a great question. I would definitely refer people to this paper, which was really interesting. And it's based on model-based estimates of mutational burden. And the argument here was that many of the mutations that occur somatically actually occur before the body matures. And they have specific information for the different tissue types, but and as the body moves and different organs mature into maintenance mode, that would slow down. So I would definitely refer the person to this paper, which is a really, I thought, very interesting paper.
Thank you. Another question is, how do you capture somatic mutation burden in healthy subjects and windows of susceptibility?
Great question. I think we're going to talk about over the next 2 days about how to think about that. Now there's more and more high throughput methods to capture non-germline alterations. I know people will be talking more about the epigenome and some other ways to capture factors in omics that change with life. So you could capture susceptibility using any of the kind of platforms that will be discussed. As long as those are measured before the environmental exposure, then that's can capture susceptibility.
Thank you. I have also a question. You talked a lot about, enriching cohorts to better really investigate gene-environment interactions. The example you gave was, as far as I understood, more on the genetic side, enriching. Can you also think other ways, what are the ways we could enrich cohorts?
Again, I would argue, if I can go backwards, sorry if this is making people dizzy. I would argue for people to do some enrichment based on age too. Again, and depending on the research question, right? Just to make sure if you are trying to understand about certain aggressive cancers that may occur at different time points, that you enrich for those. So again, I think, and this is another great article that I would that I would just refer to people to. This came out just a couple of years ago. Is that, again, depending on where your question is about cancer, but most cancers occur after 70. So if you have people in this age group, when you're looking at them, then you have to think about the somatic burden. And so I do think that it depends on what the research question is, but I think by just doing population-based sampling, you're going to get much greater heterogeneity that will make it difficult to detect certain certain interactions if your interactions are specific to germline.
There was a related question to this in the chat that was asking, if we enrich for genetics and, have carriers of BRCA1 and 2 in the cohorts, wouldn't, is the genetic effect so strong that it would overwhelm any environmental lifestyle factor that maybe has a weaker effect and may hinder finding gene-environment interactions?
Yeah, so we have not found that to be the case at all, because again, most, if you think about enriched cohorts, they're just like I showed you with the twin studies, there's a distribution, right? So people with a family history don't all have the same risk, right? Because there's modifying genes, there's modifying environment, and just like we see with the twins, there's a range of estimates. So I think but by missing this tail, so if you look at this is the estimate of average risk cohorts are in blue, and most average risk cohorts can't really look at anything at that far end of the tail.
Thank you. There's another clarification question. If the participant understood it correctly, would you advise to do cohort studies in younger individuals to investigate gene-environment interactions?
That's a great question. Again, there's always a trade-off, right? Because the rates are definitely rarer and lower in younger people. So that would be a really hard study to do, but maybe more case control studies in younger individuals. Because again, with case control studies and the ability to get tumor tissue as well as peripheral tissue, and to think about, perhaps family controls and other kinds of designs beyond maybe a population-based study. Thank you.
I think in the interest of time, we are now going to the next speaker. It's my pleasure to introduce Genevieve Leiden from Bristol University, and all is yours.
Sorry, I was stuck on mute there. I'll just check you can see my slides okay?
Yes, we can see the slides and hear you.
Wonderful. All right. So firstly, I'd really like to thank the organizers of the event today for the opportunity to speak. And it looks like a really, really interesting workshop. So yeah, my name is Genevieve Leiden. I'm a postdoctoral researcher in the University of Bristol in the UK. And for the applied example in this session, I'm going to be presenting some work from a recent study on disentangling the etiological pathways between body mass index and site-specific cancer risk using a tissue-partitioned Mendelian randomization approach. Right, so obesity is a condition characterized by excess adiposity, and it's known to causally influence higher rates of many types of disease, and it's also an established risk factor for several types of cancer. And the prevalence of obesity worldwide has risen dramatically in the past 30 to 40 years, and this is largely driven by rapid changes in lifestyle and nutrition, and this is often referred to as the obesogenic environment. So in epidemiological studies, BMI is often used to proxy excess adiposity. So BMI greater than 30 is typically used to define obesity. And it's also a very highly heritable trait. So variation in BMI has a really large genetic component. So large-scale genome-wide studies have been important in identifying regions of the genome which are reliably associated with BMI. And to date, there's approximately 900 BMI variants. And a number of studies have shown that the majority of genome-wide significant BMI variants are actually enriched in the central nervous system. So this highlights the important relationship between neural regulation of appetite, energy balance, and those homeostatic pathways. And it establishes an important role for the central nervous system in overall body composition. However, an important caveat of the genetics of BMI is heterogeneity. So there are multiple different causal pathways by which an individual may be predisposed to having a higher BMI, not all of which have the same effect on disease risk. And an interesting example of this has been the identification of a number of common BMI increasing alleles, which are associated with increased adiposity, but lower risk of cardiometabolic disease. And that's particularly the case for type 2 diabetes. And this example is really just to highlight that there are divergent mechanisms by which a BMI increasing allele may influence either a metabolically favorable or an unfavorable phenotype, depending on the context. So before moving on, I'd like to begin with a word on Mendelian randomization, or MR is what I'm going to call it. And Mary Beth gave a really nice, touched on this briefly in the previous presentation. So MR is an approach which uses genetic data to infer causal relationships in observational epidemiology. So in your conventional epidemiological observational study, there'll be a modifiable exposure, which is measured in a group of people. And then I'm going to continue with BMI in the example here, just to follow up then the relationship between your measured exposure and the outcome is evaluated. However, inferring causality from observational data is a challenge. So multiple factors could be influencing both the exposure and the outcome. And in this scenario, it's not possible to exclude confounding, reverse cause, or other sources of bias. So MR is a causal inference technique where genetic variants are used as instrumental variables, and these are used to proxy the effect of the modifiable exposure. So it's based on three key assumptions, which are similar to instrumental variable analysis. So the first is a relevance assumption, and that is that the instrument is reliably associated with the exposure. Second is exclusion restriction. So the assumption the variant only influences the outcome through the exposure. And lastly is that the instrument is independent of confounders. So the first of these is the only one you can reliably test. However, the characteristics of genetic variants mean that they make really good instrumental variables. So your allocation to your genotype is random. So you have a 50-50 probability as your chance of which allele you receive from either parent. And that natural randomization process and its consequences for inference is analogous to what you'd observe in a randomized control trial. So the analysis is very unlikely to be affected by confounding. Another primary advantage is the avoidance of reverse causation. So because we're considering inherited genetic variants, the directionality of the effect is known and the genotype remains constant throughout lifetime. So in my example, again, I'm using the genetics of BMI to instrument my exposure in Mendelian randomization analysis. So by taking all 900 genetic variants which are identified for BMI genome-wide, I'm relating them to genetic variants for an outcome, such as coronary artery disease in this example, from another GWAS, but from a similar population. And I do this to estimate the total effect of lifetime BMI on the disease outcome. And this is your kind of standard 2-sample MR analysis. And this methodology is increasingly playing an important role in genetic epidemiology studies. And our insight on genetic mechanisms brings a new level of understanding to how people are predisposed to developing obesity and how that predisposition relates to disease. So the contribution of individual tissues is a growing area of research. So in this study, we aim to identify and separate the contribution of BMI SNPs on disease risk based on tissue-derived gene expression. And in particular, we use gene expression data derived from subcutaneous adipose and from brain tissue. We next sought to compare the relationship between these tissue-grouped BMI variants and predisposition to disease. We initially developed this analysis in a 1-sample setting, investigating various cardiovascular outcomes. We've now adapted the analysis to a 2-sample setting and focusing on site-specific cancer risk. So to do this, we integrated summary-level genetic data from genome-wide studies of BMI and gene expression data derived from subcutaneous adipose and brain tissue. So these data sets comprise cis-eQTL data from both tissues. To partition the BMI SNPs, we applied a Bayesian colocalization approach called COLOC, and the variants which had evidence for colocalization, we next used these to derive novel exposures in a multivariable analysis. So, excuse me, my charger was not plugged in. So the BMI summary, to do this, we obtained BMI summary statistics which were obtained from a large meta-analysis of BMI. We identified 915 independent BMI SNPs, which reached a genome-wide significance threshold. The brain gene expression in this study was obtained from a published meta-analysis, so this is published in 2018. In this study, they combined neural tissue from 10 brain regions, and we had a total sample size of about 1,200. The adipose gene expression data was obtained by meta-analyzing 2 data sets. So for this, I used the GTEx consortium and also the Twins UK study. And altogether, we had a total sample size of about 1,200. So using regional association plots, we can look in detail at the results of the colocalization at a specific locus. So in this example, I've just plotted the ADAM-TS9 locus. So each point is a SNP or a genetic variant, and each SNP is plotted according to its chromosomal location on the x-axis. Y-axis indicates the association between the SNP and BMI, or the association between the SNP and the gene's expression in adipose tissue or in brain tissue. And these overlapping distributions here suggest that the BMI variant in this region also influences ADAM-TS9 expression in adipose tissue. So I tested this formally using genetic colocalization. So your threshold for colocalization is a posterior probability greater than 0.8. So the PPA for the association in adipose tissue was 0.97. And this is just really consistent with strong evidence for a shared causal variant. However, performing the exact same analysis using the brain gene expression data, we have really low PPA. And you can see here, there's really weak evidence for an association at the locus and ADAM-TS9 expression in brain tissue. So on the previous slide, I just highlighted one example where there was evidence for colocalization at a specific locus with BMI. But overall, the colocalization analysis was performed systematically for all 915 BMI SNPs with adipose and with brain gene expression. So out of that 915 BMI variants, I identified 140 with a shared causal variant with gene expression in brain and 86 in adipose tissue. And this was followed up by a substantial amount of further characterization. So a couple of points that are worth mentioning. The effect estimates for each set of instruments on BMI were really similar. And this is a good indication that differences that I see later on in the analysis aren't because one tissue is more strongly associated with BMI than the other. And we also related each set to additional measures of adiposity. And what's interesting here is that we see that the brain instruments more strongly correlate with waist to hip ratio and visceral adipose tissue than the adipose instruments. And this is important because this tells us that even at this level, the tissue partitioned instruments are capturing distinct aspects of biology or phenotypes related to overall body size. So the tissue partitioned MR approach is based on a multivariable model. This is an extension of the conventional MR approach I mentioned earlier, where we were estimating the total lifetime effect of BMI on a disease outcome. The multivariable MR method allows you to estimate direct effects of potentially related exposures, and this is achieved by including each exposure in the same model, but conditioned on the other. So in the multivariable model here, I've incorporated a subset of BMI variants, which had evidence for localization with both tissues, this time as separate exposures. So first developed this in a one sample setting. And to do this, we calculated genetic risk scores for participants in the UK Biobank, so the prospective cohort study. Each individual gets a score for the number of brain or adipose SNPs they had, and the scores were weighted by their BMI effect estimates. I'll briefly highlight some results obtained using this approach next, but more recently, we've adapted the methodology for application in a two sample setting. So here, instead of deriving genetic risk scores, the summary statistics for the adipose and brain BMI SNPs were incorporated into the model, but by weighting their effect estimates by their tissue-specific evidence for colocalization. To demonstrate this, I'll quickly highlight results on some cardiovascular outcomes first. So this is in the one sample setting. The plot on the left illustrates the total effect of BMI in a univariable analysis. And unsurprisingly, we observe a really strong positive relationship between genetically predicted higher BMI and each disease outcome in these cardiovascular cases. In the multivariable model, we've derived our alternative BMI exposures, and these are based on our adipose and brain genetic risk scores. We hypothesize that these tissue colocalized sets of instruments may capture distinct effects on BMI, so they may also contribute differentially to disease risk. So looking at BMI when instrumented with brain tissue first in red, that we can see that higher risk of developing disease outcomes such as coronary heart disease and stroke and type 2 diabetes, this is retained by the brain BMI effect, while the adipose effect attenuates. And then we see the opposite trend when we look at the results for left ventricular cardiac phenotype. So here the positive effect is retained by adipose BMI, while the brain effect attenuates. And body composition and fat distribution are important predictors for these phenotypes, and this appears to be well captured by the adipose exposure. So following these results, we hypothesize that the tissue partitioned BMI exposures may provide insight on the relationship between obesity and site-specific cancer. So obesity is a well-established risk factor for several cancer types, and this is further supported by a growing body of MR studies. However, the relationship between BMI and cancer is complex and site-specific. So excess fat can contribute to imbalanced hormone levels and elevated inflammation, and this increases the likelihood of developing cancers such as pancreatic, kidney cancer, colorectal, and endometrial cancer. On the other hand, there's also observational evidence for an inverse relationship between BMI and prostate cancer, and also with lung cancer. However, that one association is more likely to be confounded by smoking. So here are the results of the MR again. And as before, on the left, we have the total effect of BMI on each cancer type. And you can see there's a strong positive relationship between BMI and cancers such as endometrial, kidney cancer, and lung cancer. On the right, we have the results of the multivariable analysis. And here, I'd just like to highlight a few examples. So first, we can see that the positive effect of BMI on endometrial cancer is retained by adipose BMI, while the brain effect attenuates. And endometrial cancer is more strongly associated with obesity than any other type of cancer. And the likely pathway towards progression is related to excess estrogen exposure and pro-inflammatory phenotypes. And adipose tissue is also a major source of mesenchymal stem cells. So it may be that we're capturing this particular adipogenic phenotype, which is more susceptible to inducing endometrial cancer. In contrast, the results indicate that the positive effect of BMI on lung cancer is retained by brain BMI, while the adipose effect attenuates. And this is further supported by a sensitivity analysis looking at cigarette smoking. So it could be that we're highlighting this neurological relationship between higher BMI, smoking, and lung cancer risk. It's also worth mentioning that we observe an inverse relationship between lifetime BMI and breast cancer risk in the univariable analysis. We don't observe direct effects in the multivariable analysis, and this is because the relationship between BMI and breast cancer risk is more complicated because it varies throughout the life course. And so there's been emerging evidence of a protective relationship between childhood adiposity and breast cancer risk. And to evaluate this in more detail, we decided to repeat the entire instrument derivation pipeline, this time using GWAS results on a measure of early life body size in the UK Biobank. Doing this, we identified 56 and 53 genetic variants, which colocalized with adipose and brain gene expression respectively. The univariable results indicated that a larger body size in childhood was protective against breast cancer risk, so that was replicating the previous finding. And in the multivariable results, this indicated that the brain BMI exposure may contribute to that protective effect. So I guess really to summarize, the results of this study are really just to provide an example of how multi-omic data sets can be used in this setting to help us get closer to understand the relationship between a complex lifestyle risk factor, such as obesity or higher BMI, on site-specific cancer risk. There are some important considerations to bear in mind. So firstly, this study really benefited from gene expression data sets with a strong biological relevance to the phenotype. So we know that variation in homeostatic neural mechanisms and in adipose tissue have important effects on body composition and body size. Furthermore, the EQTL datasets were derived in bulk tissue samples, but from equivalently sized studies and actually from quite large sample sizes. So the study wasn't extended to include other tissue types right now, as the available datasets were underpowered. But there's a lot more that could be explored in this area. So initially, one aim would be to integrate summary statistics for additional cancer types. So even in this analysis, available GWAS summary statistics for some cancer types were quite limited. And that was an issue in the study, particularly for pancreatic cancer and kidney cancer. To overcome that, some of the outcome data we used was based on parental history of disease. So that was the case for colorectal cancer and lung cancer. It would also be really interesting to explore those life course specific effects in more detail. So I touched on this with the early life body size effects on breast cancer risk. And moving on, it would be really important to investigate like pre and post-menopausal effects, so integrating more age stratified effects. And beyond that, I guess primary focus of my work now will be to identify other molecular trait data and try and demonstrate the parameters around how that could be best incorporated into a causal model to address these kinds of questions. So I'd be interested in identifying additional tissue-specific omics data sets, so be it for methylation or protein expression, and hopefully demonstrating how they could provide additional context for studying these causal effects on complex disease risk. So that's the end of my presentation. So I'm happy, thank you for listening, and I'm happy to join any discussion on this work. So.
Thank you, Genevieve. This was a very interesting presentation. I'm going now to the chat and see some of the questions. One question is, what is the difference between Mendelian randomization and mediation analysis?
Yeah, that's a good point. So I guess what I've done here with the multivariable Mendelian randomization, that is a type of mediation analysis. So Mendelian randomization can be applied in different ways, and your kind of typical one is that you're just using genetic proxies to estimate a causal relationship. So that's like your univariable typical analysis. The multivariable approach I've applied here is, it's analogous to a mediation analysis. So there's a lot of similarities, and it's more like a type of it.
Okay. Then the another question is, for the Mendelian randomization, was it validated, and what is its accuracy? Did the Mendelian randomization was validated using actual exposures that contributed to the specific genetic changes?
Yeah, so if I am answering this correctly, as in how I guess the characterization of the exposure variables is what they're interested in here, and is it how accurately do we know that we're detecting like biologically different phenotypes, or so in that, to address that, I mean, we kind of followed up our instrument derivation pipeline with like a substantial amount of further characterization. And also the kind of identification of our instruments is followed quite a stringent analysis. So the genetic localization approach is quite a conservative analysis to kind of identify our kind of share genetic effects with each of the tissue types. So I don't know if I've answered that fully. I think that could relate to several different things in terms of how it was validated. I mean, if it comes to the kind of outcome results, those previous results I presented on the cardiovascular outcomes, that was in UK Biobank 1 sample analysis, but then we demonstrated that we could replicate the same findings in a 2 sample analysis. So in that analysis, using additional kind of GWAS summary statistics in the 2 sample approach. So yeah, we've kind of followed up all these results with some more characterization and sensitivities.
Okay, thank you. That's helpful. There's another question. You mentioned that you looked at gene expression in tissue. Can you explain what characteristics or expression you are referring to? The amount of fat in the tissue?
No, so these are cis-EQTL data sets. So these would be genetic variants identified in a study. So basically SNPs, which are robustly associated with variation in genes expression from, say, like a transcriptomic profile. So yeah, it's the kind of pipelines that the GTEx study would use to identify EQTL in each of their tissues.
Thank you. Another question is, isn't Mendelian randomization confounded by non-random mating patterns?
Yeah, so the kind of how you address that is that you perform your analyses in similar populations. So like all my data is from a similar population, and that's, yeah, it's referring to population stratification. So that's going to be an effect, but yeah, it's not strict randomization, but you're doing it within the context of this population.
I'm not seeing any other question. I know we're a little bit behind. So I think we should go to the panel discussion. And as was stated before, we will have 4 panelists, and we're asking each of the panelists to give a 5-minute description of what they're thinking is really important for the session. And we're starting with Catherine Mataye at the University of California, Berkeley School of Public Health.
Thank you. Thank you very much for the introduction and for the organizer to inviting me to the panel discussion. I learned a lot from the talks, and I'm studying cancer in children, adolescents, and young adults. So you know, we're being challenged here because those are rare diseases compared to adult cancers. They are also highly heterogeneous within different cancer sites. And what we see, especially looking at childhood leukemia, which is the most common cancer in children, but I'm also studying testicular cancer in young adults, is that the Latino community is definitely highly impacted by those cancers. They have either the higher risk or the higher trend, in risk. So we definitely have a situation. We have a high burden of cancer in a very specific population. So we're definitely, all the challenge that you described, Mary Beth, they're being amplified for us because we are dealing with rare cancers. And most of the time, the most efficient cancer design, cancer study design is case control study. So I was pleased to see that you kind of reframe the usefulness of some of those designs in your presentation to not negate the advantage of working with case control design, and we can still learn from those. This being said, we're still very challenged with this sample size, and I hope we're going to talk about this later in the next sessions. But I was hoping maybe to have some fresh ideas of how we can get around issues around sample size. Can the statistical methods help us to really alleviate that? Now, the positive side of working with childhood cancers is that we do provide a very good model for looking at developmental origin of cancers. And our team has been successful to obtain blood specimens from at the time of birth to be able to have, omic characterization in terms of genetics, epigenetics, metabolomics that is respecting the temporality, and we can do that. But again, case control studies are most of the time challenged, so we were able to compensate that with this type of specimen. But again, I'm really excited to hear about the next session, because we are far from being, having integrated all those layers of omics that we have characterized at the time of birth for development of cancer later in life. We're still not there, and so I'm really looking forward to that session to see how we could approach that. Lastly, I'd like to say that we, again, because we are looking at the in utero, window of susceptibility, we were able to access fetal tissue to really characterize epigenetic profile in normal tissue before we could even look at tumor tissue in our leukemic patients. So having both the normal tissue and cancer tissue is definitely important, but can be very challenging to obtain in some instances. So I'm looking forward to learn more about how I can improve my studies to take advantage of all those omics in the context of rare cancers.
Thank you. Our next panelist is Sophia Wang from City of Hope Comprehensive Cancer Center.
Hi, Ricky, and thank you everyone for having me on this panel. It's a real honor to be here this morning. I guess almost noon for everybody else. So I guess I would like to build off of what Dr. Terry has talked about earlier, because I think that the issue of this will probably come off as more of a plea to as epidemiologists, because I think that the issue of study design is really critical, and the inferences that we make based on the study design is really critical. And it's a really exciting time now because we have so many of these omics technologies, and we have GWAS that are well underway, and we have exposure data, and we have gene expression array, all of which we are hoping to incorporate in, from my perspective, the work that I do in non-Hodgkin lymphoma. And I think a phrase that we often hear is we can quote unquote get tissue. And I guess I want to bring it back to the study design and really consider from, go back to Epi 101, and consider the generalizability and the inference that we make from these efforts to quote unquote get tissue, because there's great opportunity from what we've all been building for the last 2 decades, but there's also some challenges. And I guess I'd like to bring it more to the forefront on those of us who are launching new studies. Maybe the targeted approach is the way to go, as Dr. Terry has discussed or brought up in her talk. Maybe there's ways in the back end statistically to weight our sample sizes or populations to understand the inferences. But just to highlight, some of the work that I think many of us on this panel have done, but when we're part of a cohort or a case control study and we're attempting to quote unquote get tissue, we have to consider the biases that our original population started with. So we already know that for for what I study, non-Hodgkin lymphoma, we have many different subtypes, and some subtypes are more aggressive than other subtypes. And so when we compare our survival curves of our more aggressive subtypes to that of the SEER survival curves, we already know that our population or our case population, it looks a little bit different, maybe though there is already a survival bias. So we have to be careful in terms of what inferences we make based on the associations, or if we're hoping to do survival-based studies leveraging these samples or this case population. But when we talk about what tissues we can acquire, I think it's a little bit more complicated, right? So already we're asking who among our case population, who is a subset of the generalized patient population, even had surgery to have the tissue obtained. And then of those who even had tissue or surgery for where tissue would be available, we have to consider who consented to have their tissue. And of those who consented to have their tissue, we have to consider which hospitals have those tissues and are willing to provide those tissues. And then once we get to that subset, you can see there's already several layers. We have to consider, once we acquire the tissue, how many of those tissues are adequate? Like, is the integrity of the tissue still, sufficient for the molecular assays or gene expression arrays, largely, that we would like to do? And then how many of those tissues that have adequate tissue have sufficient quantity? So you can see that there's several different layers, and what we get at the end of the day is extremely important, but that comes with epidemiologic considerations and what inferences that we can make. So I guess my plea is more, I think we have to pay attention as we launch studies, maybe as a focus study of trying to get, really keep the integrity of the case population intact, rather than, doing a more generalized call for quote unquote getting tissue from wherever we can, which is, I think, what we've all done to date. And a plea to maybe our biostatisticians to help us, understand what inferences we can make based on these tissues, because I think they hold incredibly important information. I think that this work needs to grow, particularly in understanding the molecular changes along with the GWAS, along with our exposure data. But I think there's some careful consideration that needs to occur for us to make the correct inferences. And with that, I will stop.
Thank you very much, Sophia. Our next panelist is Peter Kraft from Harvard University, the A.T. Chen School of Public Health.
Great, thanks. So I wanted to talk about two topics which I think build on things that have come up already. So we've heard a lot about the importance of study design. I want to talk a little bit about the importance of appropriate statistical analysis. And to sort of build off of what Sophia was just saying, I don't know that I can solve that problem, but I could definitely point to the importance of acknowledging it and documenting where are we getting our samples, what's the timing of our samples, who are these people anyway. So those are my 2 points. First, I want to talk about aligning our statistical analyses with scientific goals. And second, good practices in data documentation and harmonization from the standpoint of modern, fair data sharing practices. So making data findable, accessible, interoperable, and reusable. So I'm just trying to get the ball rolling on these questions. I'm sure they're going to come up in the other sessions as well. So on the first topic, our scientific goals should drive our choice of analysis. Now, this requires careful thought. Not only do we need to be clear about what our biological hypotheses are, but also we have to be careful because vague or confusing terminology can get in our own way. So when we talk about biological interactions, for example, we're talking about something in a completely different domain than statistical interactions. So statistical interactions are defined in a wonky way as effect measure modification or departures from additivity on a specific scale. And in and of itself, there's nothing about departures from additivity on the log odds scale or on the absolute risk scale that need imply biological interaction, especially when we haven't specified what that mechanism of interaction is. So for example, a statistically significant multiplicative interaction term from a case control study, from a logistic regression of a case control study, doesn't say anything about what's happening between a gene product and metabolite at a cellular or tissue level. And conversely, two factors may interact even though you don't see a statistical interaction. So we need to think through our implications of our mechanistic biological models for the kinds of patterns of associations we expect to see in our population-based studies. And this will inform which analyses we want to perform and how we interpret them. So on the second topic, data harmonization also should be driven by our scientific goals. And good data documentation can preserve our ability to use data to address multiple questions using different approaches. So, for example, many consortia looking at the association between lifestyle risk factors and cancer risk involve many different studies that have assessed these factors using different, very heterogeneous questionnaires. So in the past, when we sort of pooled data and created a single harmonized data set, they've often transformed study-specific data to a common standard, which is usually defined by the least common denominator. So that's how we end up with big pooled data sets where we have the ever never smoker, even though there are many studies that have very detailed information on quantity packs per day over the life course. So going forward, it should be possible to share data while still preserving the level of detail that at the individual study level. And this will enable more refined analyses. And it'll also enable that scientific goals can drive whether or how data are harmonized. And researchers will be better able to communicate the potential drawbacks from their harmonization choices. So as one example, the Population Descriptors Working Group for the NSGRI's PRIMED Consortium has developed a data model that allows each study to preserve detailed information on race and ethnicity and document how that was collected. So although some studies may have collected race and ethnicity data using familiar, very broad OMB categories, more detailed information is indeed available on others. And it'll be important to use that information in order to disaggregate those heterogeneous categories like Hispanic or Asian American Pacific Islander. And I want to stress that recognizing and documenting heterogeneity is not only important for projects involving multiple studies. Measurement accuracy within a given study may be correlated with other factors. For example, there may be more air pollution monitors in wealthier neighborhoods than in poor. And this can lead to an apparent effect measure modification, which is strictly due to this differential measurement error. So again, I think both when you're thinking of what analysis should I run, how to interpret my analysis, and how do I set up my data sharing framework, those should really be driven by our underlying scientific goals.
Thank you, Peter. Our fourth panelist, Catherine Hoyo from the North Carolina State University.
Thank you for the introduction and to the organizers for inviting me to this. It's a really informative workshop and the opportunity to learn and share some of my thoughts in the omics work that we do. So to do this, I thought it would be most informative to talk briefly about what our research focus is on cancer and the environment, and then the challenges we face, and then talk about the omics approaches we use and how these approaches may contribute to early detection and of exposure and exposure effects, or even outcomes such as cancer. Maybe end with the challenges that we are currently facing, and we have no idea how we're going to solve them. So our research program is overly focused on the understanding of the effects of environmental contaminants across the life course on susceptibility to cancer risk and mortality. And this is really for risk stratification and early detection to reduce health inequities. So our program does gravitate towards those cancers that disproportionately affect ethnic minorities. The most time and cost-efficient design that we do use is for these investigations remains the case control study, where participants are selected from the general population and in cases of cancer identified, and then compared to non-cases with respect to exposure. Although nesting case control studies within these large cohort studies that Dr. Ellison has been talking to us about, it has been, and as both Dr. Ellison and Terry have said, many existing well-phenotyped cohorts with really long-term follow-up have very limited specimens that can be used to measure environmental exposure. And they tend to have very, very few ethnic minorities. And so they tend to be extremely underpowered. It makes it very difficult to make any to draw any conclusions from this. So a major obstacle in interpreting the case control data that we do have has been the, as we think about cancer outcomes, has been, as we all have seen, the half-life of many of these emerging contaminants, such as the Gen X story, for example. Either the half-life itself is very short, or the chemical structure is not known, or parent compounds or metabolites are not known, such that the measurement of contaminants make cause and effect very difficult to infer. And this temporal ambiguity between exposure and outcome is especially difficult when we are trying to evaluate the relation between exposure and cancer susceptibility. So to overcome this type of obstacle, several groups have turned to epigenetics, which is a means by which genes respond to their environment in order to identify stable, environmentally responsive epigenetic marks that mediate relationships between the exposure to the contaminant and chronic diseases such as cancer. And like many of us in this workshop, the epigenetic marks we investigate the most is cytosine methylation marks, which would probably distinguish between or differentiate between those exposed and the unexposed, or the cases from the controls. And we tend to use available platforms like RRB, the reduced representation by sulfide sequencing, or the methylation arrays. And a major challenge has been the platforms that we have very low coverage. Cause and effect is very difficult to infer. And sometimes because you necessarily use tissues such as blood and saliva, which are accessible, they tend to not have the same problem. So we have developed imprint marks that are these are methylation specific, that are parent of origin dependent cytosine methylation marks that control genomic imprinting. We used to have about 10 that were, about 25 that were known, and we have developed the rest of them now. And we are working with Illumina to put them on a platform. But what remains in this are three major challenges, right? One of those challenges is, how do you actually analyze this data when you're taking people from different ethnic groups? How do you pull that DNA? And then how do you then, as we all know, these imprint marks are established very early in gastrulation. So how do you then look at marks that are beyond that period of beyond during the postnatal period? Because even though we know from the Barker type work that early period is important, there are other periods that are just as equally important, and we haven't identified marks that are stable enough. So I'm going to stop here.
Thank you, Catherine. Now we're opening to the overall discussion. So if anybody in the audience has questions, please put them in the Q&A chat. And I want to first start asking the speakers and panelists if there's anything they would like to respond to that has been asked before, and they would like to share some insights. Mm-hmm. If not, Catherine, if not, there's one question that came right at the beginning that asked about what is our progress on gene environment actions. We're talking about this for a longer time, and what can we really show so far? And a related question to this is, should we call it gene environment interactions or environmental times G interactions, or E by G? Who likes to take this question?
Well, I was actually about to address the second point, the E by G. So yeah, yes. So, often we think of it like, is the effect of the gene different depending on your exposure background? But of course, you can turn that question around. Are there particular genotypes who are particularly susceptible to environmental effects? So it's useful to phrase it both ways. And again, that gets back to sort of what's the scientific goal. If it really is, I think, as Mary Beth pointed out, we want to make sure that we're doing something that works for everybody. So if there is a subpopulation that's particularly susceptible to an exposure, we want to be able to identify those. So that it could be that there are folks who have pathogenic variants in a particular gene, and they're particularly susceptible to certain exposures, and we'd want to know that. I will also briefly touch on the first question. What do we know about G by E? It sort of depends on what you want to know. So in terms of understanding biological mechanism, I think there are a handful of examples where we have a pretty good understanding of what both G and E are doing and how they're interacting biologically and why that leads to the patterns we see in the general population. That short list may not have changed much over the last 20 years. But on the flip side, in terms of if you're thinking as a risk modeler or identifying subpopulations who are at high risk of disease and might benefit from interventions, we know a lot more about how inherited genetics and environmental exposures interact or jointly affect risk, if you will. And that's allowing us, the potential for more targeted interventions.
So I think, Peter, you have kind of combined, what we defining gene environment actions here a little bit. Are we looking at a specific genetic variant, or are we looking more of genetic susceptibility? And I think we can even spin this broader and divvy up in different ways the genetic side that we haven't probably done in them so far. And I see Sophia raising her hand. We can't hear you, Sophia.
Okay, can you hear me now? I apologize. Yeah. Okay, so I would say, Ricky, I think you probably have some successes that you could be shared with the group. But I guess I feel hopeful. I know we've been doing this for a while. I see Nat has his hand up. I think a lot of it depends on the study design, right? Like Dr. Terry said. So for many of the cancers, for G by E, we do require large sample sizes. So I think some of the cancers that have been able to accumulate GWAS in large sample sizes are a little bit or much more ahead than, say, the cancer that I work on, which is lymphoma. But even then, I do feel hopeful. I think that Dr. Terry has a very good point when she says perhaps we need to have a more targeted approach. The way that we've tried to approach it is looking at those who are particularly susceptible, maybe with autoimmune conditions. I think, really focusing on for, some of the cancers with rare, that are more rare, focusing on these more susceptible populations is probably an approach rather than, taking the entire population, because we'll never get to the fifth, well, maybe not, I shouldn't say never, but it would be very difficult for us to get to the numbers that, say breast or colon cancers have accumulated. But I do see this as hopeful. We're finding, many cancers have discoveries on GWAS, and I think we need a little bit more time, but I think we've learned a lot based on the GWAS that we've done. And I think we're still learning, how to, what kind of G by E. Are we looking at SNP level? Are we looking at PRS level? Are we looking at biological level? I think there is much more that can be done that we have yet to discover.
Thank you. Next, Nat Rossman has his hand up. I don't know if can we unmute you?
Can you hear me?
Yes.
Hi. It's a great meeting. Hi, Ricky. Haven't seen you in a while. I want to make two quick points. One, migration studies and secular trends in populations unequivocally have shown that cancer is being driven primarily by the environment. Genetics play an important role. High penetrant genes in a small part of the population. Low penetrance genes play a role. This is clear, but the change in rates and populations that migrate are just so striking that I think we really need to make sure that we have the right perspective on what's driving risk, and think not just at the individual level and trying to identify susceptibles, but think at the broad public health level, and in the context of trying to identify what are those exposures, broadly defined as everything that's not inherited, that are driving risk, and to try to intervene at the population level, either through regulation or through prevention guidelines to reduce the risk of disease. In addition to risk prediction models that try to identify the so-called susceptibles and to either more intensive counseling or screening. I mean, a high proportion of the population gets cancer, and I think that we can have a big bang for the buck by looking at studies of the broad population, in addition to focusing on higher risk populations, that we can learn a lot of what's needed to learn that we can benefit from by studying unselected populations in very good studies. So that's sort of my one fundamental point that I wanted to say. Two, if we agree that the environment is driving much of cancer risk, then, as a number of speakers mentioned, particularly at the beginning, it's really critical to try to identify those exposures. Again, apart from the holy grail of looking at gene-environment interactions, which many of us do, and I do as well, it's important to characterize those exposures in and of themselves as main effects, and that's been what has been elusive in environmental epidemiology. While there are some exposures, some lifestyle factors that we can capture reasonably well, many environmental exposures are much more difficult, more challenging, and hopefully technologies like metabolomics, which is really, it's called metabolomics, but it's just the analysis of small molecules, which can include parent compounds, not necessarily metabolites, measuring exogenous exposures, and application of those kinds of technologies may actually help us, particularly in cohorts with repeat sampling, ideally cohorts that have younger people, to try to characterize the endogenous milieu, but understand what's driving that endogenous milieu, particularly those exogenous factors that drive cancer risk, that drive most of cancer risk in most populations. So I think exposure assessment, really good exposure assessment with minimal measurement error, captures at the right times in life course is one of the one of the key focuses that we should have in our thinking.
Thank you, Nad, for sharing this. I see Catherine Hoyo has her hand up.
Actually, I was going to say a lot of what Dr. Ruffman has already said, that what I would like to say is that the environment does contribute substantially to disease risk, and how we define that. And I think we have enough technologies now to help us define it. There are some of us who really think that we can learn a lot from the DNA that many of us have in our freezers already, in order to try and capture what environmental cues are related to these exposures as intermediates to disease. But I think that it would help the research community if we actually focused on environmental exposures. And I think the work that has been done in metabolomics, if we could combine that, say, with DNA methylation, it would go a long way in trying to sort out the effects of the environment. The last thing I was going to say was that the social stressors that are very difficult for a lot of us to measure are being measured by social epidemiologists, and they seem like they interact in ways that we still don't understand with the environmental contaminants, or they co-occur with these environmental contaminants. And it has been a very difficult thing to do, to try and combine. These are different scales that we are measuring the contaminants in, and the social stressors, and just trying to combine that and seeing what disease susceptibility we can find is going to be critical moving forward.
Thank you, Catherine. I see in the chat questions related to study design overall. So, you know, the panelists, if you can look at them specifically, but also kind of the overall question, you know, in terms of window of exposure, long latency, what would you like to share with the audience?
What's the question? Sorry.
The question is broadly about study design issues. You know, what is a window of susceptibility, long latency? How do we measure environmental exposures? It's a broader question, and then there's two specific questions in the chat. One is about a retrospective design. You know, how far should a person in a case control study go back to assess exposures? 5 years, 10 years. And the other one is, you know, do we have recommendations about the design of the architecture of a multi-omics research consortium? Anybody like to share thoughts about what they learned about study designs?
Can I say something briefly? I think that case control studies will remain an important part of what we do as epidemiologists. I don't think we can run away from that. But I also think that the challenges we face, that where the exposure that we're talking about is measured at the time of disease diagnosis, and the fact that we really have very little to go by going back to address that issue of temporality, means that we have to find intermediate markers that are going to tell us this person was exposed, and understand that these intermediate markers we're looking at are actually stable, because there are some epigenetic marks that necessarily need to respond to environmental cues, including disease cues. So the question is, can we find those stable marks and demonstrate that these are stable, and then use those as surrogates for past exposure? But I don't think we can run away from case control designs, whether we are talking about population-based case control studies, or we are talking about those that we nest within cohorts. We still need to be able to do that.
Thank you. Anybody else like to share some thoughts? I see Sophia has her hand up.
So I don't know that this is going to answer any questions, but I'm just reading through the chat now, and someone asked, you know, what is the optimal time? And I guess of exposure assessment, if they're planning a case control study, and I guess I would go back to what, Dr. Kraft said. It depends on your scientific question. And I think that's where the rub is, right? Because those of us who are doing case control studies have a very specific question and specific exposure that we're targeting, and yet when I was speaking earlier, I was looking at this, landscape of cohort studies, the All of Us, or these new studies where they don't necessarily know. You have to anticipate all the questions that you may want to ask downstream. I think it's a very different landscape to try to merge together. So, I do think the research question matters. I think trying to anticipate what research questions you want to answer matters. And that's sort of, it's based on what we know today. It's not an easy question to answer.
Thank you, Sophia. I see Catherine Metayero has her hand up.
Yes, I like to share some of our experience working with cancer in children, so that the time the latency is shorter. We're looking at leukemia, so the peak for leukemia is 2 to 5 years old. So we have a little bit less time to cover. But the approach that we've taken, because there won't be one perfect one, but we took a multi-tier approach where we do the traditional interview of the parents to gather as much self-reported data, which is still useful. We did a lot of GIS, geolocation and linkage to the residential history that we have for where the mother was living when she was pregnant, when the child was in the early years of life. So we have, then geolocation information. What we did was very interesting, and I know that groups also, we've been working with groups from NCI, is to collect dust. Dust is a very interesting media where you actually can identify persistent compounds that have been in your house for decades, and we were still able to measure, dioxin. We're still able to measure contaminant that have been banned for the longest time. So we've worked with those type of media, the dust, and what we've done to kind of answer the question, how far back can you go to identify chemicals in your home that would be stable? We went back and did a second measurement, like 3 to 7 years after the first one, to understand the stability of those compounds in your home. And you'll be amazed that it went down, but it was still, the correlation was still high. And then what we did is that, because we're dealing with children and we feel that we could not collect blood for the controls, the healthy children, we could not ask to have blood sample taken. So most of our samples are from blood samples are from the child who had leukemia, and we collected blood at the time of the diagnosis. And so we also compare those dust level, the environmental burden of chemical, to the blood level of those chemical. And we did this analysis for PCB, PBDs, and we find a correlation that is substantial between what we find in the home and what we find in the blood. So to go back to the question of the, that we had, there are many ways that you can try to identify the persistence of the chemical that you're looking at, and be creative to do this type of sub-studies where you're going to have specific environmental exposure sampling that you can kind of correlate with the the omics side of your work.
Thank you, Catherine. I see Mary Beth has her hand up.
Thank you so much. I absolutely agree that it's the environment with a capital E. The twin data in particular, if your identical twin gets cancer, you're actually more likely not to get cancer. So it's absolutely the environment. But I would say, and I love that people are talking about case control studies again, that there is a very real challenge in epidemiology to get consistent results when we're dealing with ubiquitous environmental exposures that people are exposed to every single day. So outside of the occupational setting, I do think we need to think about ways to really not only measure the environment more accurately, but really think about the time in life and susceptibility, whether that's susceptibility from germline or susceptibility from acquired mutations. And most mutations are, most cancer genes are altered through acquired mutations. It's only the minority that are through the germline mutations. So I do think it is environment with a capital E, and we need to think about that all samples are not equal. And as Dr. Wang said, we need to really think about not just taking tissue from wherever we can find tissue.
Yeah, that's a good point. Thank you. Catherine, do you still have your hand up, or is it?
Yes, I like to bring another point is that in terms of our understanding of the molecular basis of carcinogenesis, a lot of emphasis is being put on mutation, but I think we also have to understand that the relationship between the cell and the stroma is very important, and that the impact of the E on the healthy stroma that can really influence the status of your cell becoming tumor cell or tracking back to being a healthy cell. I think that has been maybe underlooked or maybe because of the difficulty to address this aspect in epidemiologic studies. But I think we have to understand that environmental factor will affect the stroma that is nurturing the healthy or tumor cell, and I think that's a very important component that is really hard to track down, but that we should not forget.
So that question related to that probably is, what kind of specimens, tissue, blood, urine we should collect in general, because you talk about now we should also look at the stroma, the tumor microenvironment, basically.
Well, again, I think it's cancer site specific, but I think it's adding a layer of complexity when you want to design your study, if you really would like to assess this.
Yeah. There was a broader question, if we could build something like the Cancer Exposome Atlas, how should we do this? Compare, we have the Tumor Genome Atlas. Is this possible to do something like that? And how should it look? Do we think it's possible or not? Catherine, you have your hand up. We can't hear you. I don't know, you look not muted. Still can't hear you.
Okay, how about now?
Yes.
Okay. So I was thinking about looking at it. I think that the developmental window is going to be important in actually thinking about susceptibility. So if we were to look at a human as they lead their lives longitudinally, I think understanding, because a lot of risk factors are shared within certain age bands, not small bands, but like really large age bands. And I think that if we could understand exposures and characterize them for age bands, we probably could get at least be able to chop the population into different susceptibility periods and be able to answer some of the questions and be able to intervene a little earlier. So I think there's a way in which we can do it longitudinally, but I can also see how we are structured to look at a disease now in a case control fashion, and therefore you kind of have to look at a disease at a time, a specific cancer at a time. But I really think that the way we're going to maybe make some headway is going to be looking at it by susceptibility window.
Thank you, Catherine. I see that Doc Walker has his hand up.
Yeah, I just wanted to speak to the feasibility of the Cancer Exposome Atlas. I mean, I think this is an incredibly important initiative and something that we do need to be thinking of as we continue to develop these resources that are leveraging exposomics and multiomics to study different cancers. I think the challenge that we've currently faced as a field is there's not a lot of standardization or consensus around the exposome methods or really what the exposome even is. But there's been a lot of efforts to move towards operationalizing that definition and designing ontologies that allow us to really link across different measurements and different scales of the exposome. A lot of the methods that we've established and others in our projects has really tried to emphasize this idea of cumulative exposome databases and kind of building this as a resource. And I think with all of the excellent cancer cohorts and the resources available, this would be actually a fantastic application of the exposome and really a way to demonstrate the power of these exposome disease atlases.
Thank you. And I also see that Dean Jones has his hand up.
Yeah, I was really just going to echo what Doug said, but also say to add two things. One is from my perspective, it's not only possible, it is essential. It has to be done. And secondly, from my understanding of the origins of the Cancer Genome Atlas, when they started, they didn't know how to do it either. And so I don't think we should let that be a barrier to getting started.
Thank you. Nat, we have 1 minute.
Okay. In less than 1 minute. We're trying to contribute to at least the beginnings of an exposure, a cancer exposure atlas by doing a series of cross-sectional studies for several dozen chemicals, looking at populations exposed to a wide range of those chemicals, characterizing exposure as well as we can meticulously, and then collecting samples in a way that they can be analyzed for a wide variety of omic technologies, and then having those samples analyzed on the same platforms with the same groups. A similar work could be done experimentally, but having a group of people who do these kinds of studies in humans and people who would do experimental work to talk together, I think could really be the beginning of developing an atlas, so that for a given exposure, whether it's for organochlorines or chlorinated solvents or PAHs or things that are possible carcinogens, but we're not sure, we could develop data on what kind of signals, what kind of metabolomic effects downstream we would see, methylation effects, transcriptomic effects, that almost to the point of developing certain signatures, and that could be extremely useful applied to, especially to prospective studies. So I think, Ricky, it's a great idea, and different groups are sort of working on it, but there really doesn't seem to be one big umbrella where people are talking to each other and trying to coordinate.
Thank you. I really appreciate everybody's comments. I have the pleasure to wrap this session up. First of all, thank you to the two speakers to give this very insightful talks, both on the overall study design, what we need to consider, as well as some individualization approaches, and all the panelists to providing their insights about this complex and difficult question we need to face, but really need to make progress on how to really integrate the environmental and genetics and multi-omics approaches to really move our field forward. It's clear that the study design, the biospecimens, the timing of when we selecting is critical for us to make progress. And it's critical to think about the research questions we're trying to address, which of course is for case control studies, very different often than for a broader cohort study that tries to envision a lot of questions in the future. And it sounds like, we should be encouraged to really think, to deep dive into better understanding the environmental risk factors with all the new technologies that come now at our hands. I think the future is very bright for a lot of our investigations, because there's been so much progress being made on the technology side, and we have strong epidemiological skill sets and tools in hand to really make progress in this area. So I'm excited for the next decade for us to come to do really important research. With that, I'm giving it back to the organizers, Kim and Leah.
Yeah, thanks so much, Ricky. I think that was a great first session. We're gonna take a lunch break now. And we're going to try to resume at 1:35 Eastern Time. So thanks, everyone.